Welcome, everyone. Thank you very much for this opportunity to present my work, which aims to dissect cell-cell communication and the perivascular niche in melanoma. So in our lab, we are interested in intratumor heterogeneity in melanoma, and by using single-cell RNA-seq, we were able to identify 7 distinct populations. I'll just put here 7 distinct populations of malignant cells. And this cluster number 4 attracted our particular attention because the transcriptomic profile of those cells is very similar to pre-empty cells that has been previously described as the first neural crest-committed cells that emerged during development, before even cells delaminate from neural tube and migrate to colonize different sites of embryo and give rise to different organs. So based on this, we hypothesized that those cells may, in fact, function as melanoma stem cells, and in consequences that melanoma grow may be hierarchically organized. And to confirm this hypothesis, we established a confetti mouse model of melanoma, and we analyzed by confocal microscopy clones. And we also engaged into collaboration with Ben Simon from Cambridge University to establish mathematical model of how melanoma grows, And this model predicted that indeed melanoma are hierarchically organized, and this cluster number 4 population is predicted to sit on top of this hierarchy. So then we wondered if those cells are randomly distributed in the tumor or there is a specific location. And for this, we performed different spatial transcriptomic assays, and we were able to show that this population is predicted to be in close proximity of blood vessels. We also confirmed this by more targeting approaches in mouse and human samples, which altogether suggest that there is something in this environment that is important for melanoma differentiation and stem-like phenotype. So we were wondering which exactly component of the perivascular niche is important to keep this phenotype. And to answer this question, we opted for a reductionist approach, in which we co-culture our melanoma cells expressing doxycycline-inducible M-cherry H2B model to follow the proliferation of those cells with different stromal populations, such as endothelial cells, fibroblasts, and pericytes. And as you can see from the quantification, each stromal population that we tested were able to stimulate melanoma growth, but endothelial cells have the strongest effect. And that was also confirmed in vivo by co-injecting endothelial cells together with melanoma, which accelerated tumor growth, and also in 3D settings where we added endothelial cells to melanosphere, which dramatically increased the volume of the sphere. Our single-cell RNA-seq sequences on co-culture also shown that there is increase in stem-like signature and neural crest signature, where decreasing melanocytic signature, which altogether again suggests that there is the differentiation process going on in co-culture. And when we look at top 3 upregulated genes, we see ID genes, which stands for inhibitor of differentiation, which is in line with our hypothesis. So to see if this effect of endothelial cells is mediated by physical contact or paraclinic signaling, we collected conditioned medium from endothelial cells, and we treated our melanoma cells with M-cherry reporter with this conditioned medium. And as you can see from the quantification, that was sufficient to recapitulate the effect of co-culture, suggesting that paraclinic signaling is driving this effect. And because endothelial cells are known to communicate with the environment via extracellular vesicles, we also separated conditioned medium fraction by ultracentrifugation into EV-depleted and EV-enriched fraction. And again, we treated our melanoma cells with both fractions. And we could see that in EV-depleted conditioned medium, there is less proliferation than EV-enriched fraction, suggesting that EVs may be implicated in melanoma reprogramming by endothelial cells. We also use the tools that was previously developed in Professor Varinen lab, in which we can overexpress Curie recombinase that is packed into extracellular vesicles in sender cells, so in our case, endothelial cells, and we co-culture them with melanoma cells, which express log-stop-logs TD tomato cassette. So cells that uptake those EVs should turn on TD tomato. And as you can see here, this is the case in our co-cultures. We can then isolate those cells and profile their transcriptomic, and we could see the enrichment of ID genes, showing that EVs carries some molecules that induce this melanoma reprogramming. And to dig a little bit into molecular mechanism of this, we decided to do proteomic analysis on EV that have been isolated from conditioned medium, as well as cells in direct co-cultures. And to select our hits, we decided to take proteins that are present in EVs and at the same time upregulated in melanoma after co-culture, but also downregulated in endothelial cells. And we could see that there is basically only one protein matching all those criteria. And this protein was neogenin, which is a receptor for axon guidance molecules, which may also suggest that it's involved in melanoma innervation. And it's well known that nerves and vasculatures are interconnected, not only in the tumor, but also in the healthy tissue. And this is also supported by our observation that neogenin is mostly enriched around blood vessels at the perivascular niche. So we decided to check if our melanoma tumors are innervated. And for this, we use neurotracer, WGA, which is conjugated with fluorochrome. And we injected this into the tumor. This neurotracer then is transported along axons to soma of neurons, which is located in dorsal root ganglion in the spine. We collected dorsal root ganglion, and we look under the microscope if we can find any positive neurons. And based on this experiment, we can conclude that indeed those tumors are innervated. So we established also DRG-derived neuron in vitro model, and we co-culture those neurons with melanoma cells and endothelial cells, and we could see further increase in proliferation when we added neurons, which was not observed after we added any other stromal cells in the co-culture. So because it's well known that neurons may affect endothelial cells biology during development, we were wondering if this is also the case here. So we collected conditioned medium from neurons, and then we pre-treated our endothelial cells with this conditioned medium, and we put endothelial cells in co-culture with melanoma cells expressing our proliferation reporter. And again, we quantified the proliferation by FACS, and we could appreciate that pretreatment of endothelial cells with neuromedium further increased their capacity to reprogram melanoma cells. And that was not the case for any other stromal population. So right now we are starting also to investigate how neurons are reprogramming endothelial cells, and the very preliminary results is that when we take this endothelial cells pretreated or not with neuromedium, we could already see that those endothelial cells that have been pretreated secrete more vesicles, which was confirmed by NTA analysis as well as protein content. So right now, our working model is that nerve endings communicate with endothelial cells at the perivascular niche, and this increase the secretion of EVs by endothelial cells, and those EV contain neogenin receptor, which help to reprogram melanoma cells into more the differentiated phenotype, and this also increased the responsiveness of melanoma cells to neurons, and in consequences further reprogram them into melanoma stem cells. So with this, I would like to thank my supervisor and the whole lab, as well as the funding, and also Champalimaud Foundation for a travel grant, and thank you very much for your attention.